today's topic is genic balance in Drosophila. Before going to detail the topic, you must know the Y chromosome and the male development. And we all know the current analysis is going on the Y chromosome because it has numerous genes and the regions and have the potential genetic function also. Some genes are within or without the homologous counterpart seen on the X chromosome, and they are present on the both the ends of the Y chromosome, that is known as pseudo-autosomal regions. So, pseudo-autosomal regions, PARS, and these genes are similar in X chromosome, which is seen during the synapses and recombining of meiosis. And other than that, 95, only 5% are like this, and 95% of Y chromosome are NRY, that is non-combining region of Y. This non-combining region of Y has two types of genes. Functional genes, that is euchromatin, which is responsible for the sex determining gene SRY, and this SRY is present means it is the male development. It is absent means it gives rise to female development. Okay, and it is seen in the Y, that is SRY is seen in the testis determining factor TDF. Okay, and it is euchromatin. It's a true chromosome to produce a sex. And another non-functional genes are known as heterochromatin, and this SRY is absent in human, it is absent in X chromosome. So due to the absence of SRY, the sex is female and it develops into the female, and it is absent in the X chromosome. And this SRY is seen in the testis determining factor TDF. So this is an explanation for the previous slide. I will explain once more. The Y chromosomes are pseudo-autosomal regions similar to the X chromosomal regions seen in the synapses and recombining during meiosis. 95% of Y chromosome are referred as non-combining region of Y, NRY. So the NRY has functional genes euchromatin, within it SRY is present, and non-functional genes are heterochromatin. In human, the absence of Y determines the female development, and this SRY gene is absent in X chromosome. The gene product of SRY is the testis determining TDF, that is TDF factor, that is TDF, present in the adult male testes. Now we are going to see the topic in detail, genic balance in Drosophila. So this genic balance mechanism of sex determination in Drosophila was first studied by C.B. Bridges. He took, that is, we all know the sex here, we took the letters as X, 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 Y type. The 2X denotes the femaleness of the fly, and the XY denotes the male, and here the Y is responsible for the fertility of the male, not the maleness. Only the autosomes are going to determine the maleness of a fly. Okay, and you can, and by determining the maleness, femaleness, that is X ratio, X is to A, that is the femaleness is governed by X, and the A, that is maleness. So female is to A, male. It is determined by the ratio. So we are determining the sex ratio. So it is denoted as sex index with a formula of sex index is equal to number of X chromosome by number of sets of autosomes, that is X by A. And if there is any change in the ratio, it leads to the change in the sex also. So for that we going to see an example, that is the from the example we get and results can be obtained. So Bridges had crossed the triploid female fly and a diploid male fly. So here the triploid female is crossed with diploid male. So the parent has 3A plus triple X, and the female and the male parent has 2A plus XY. So the gamete formation in the triploid female has two types, that is first type is 2A plus XX into A plus X, this one type. Another type is 2A plus X plus X, A plus double X. These are the two types of gamete formation from the triploid female parent. From the male diploid parent, A plus X one type and A plus X, Y, and A plus Y another type. So now we going to use it in a Punnett checkerboard. So this one is, this side it is female and this is considered as male parent. So we are writing the first genotype, that is this genotype 2A plus XX, and here it is one A plus X. Next you are writing this one, that is this 2A plus X, and here you are writing this one 2A plus Y, and next this one that is A plus this one A plus XX and A plus X. So now what we going to get in the checkerboard, that is 2A plus XX, that is 2A plus XX plus A plus X. So 2A plus A is 3A plus double X plus 1X, so triple X, okay va? And here 2A plus 1A, it is 3A plus XXY, so XXY, so it is a triploid female, and here it is triploid intersex. Next 3A plus XX, it is a triploid intersex, and 3A plus XY, it is a super male, and 2A plus triple X is a super female, and 2A plus XXY is a diploid female. 2A plus XX is a diploid female and 2A plus XY is an diploid male. So these are the offspring we are getting from the cross. And the interpretation of the ratio from the cross, that is A, that is X is to A, that is X is the X chromosome, that is the female is to male. The X is from the female chromosome and A is from the autosomes to denote the maleness. So if the ratio is 1, it is normal female. Greater than 1 is female, just female. And the ratio is 0.5, it is a normal male. Less than 5 is a male. And 0.67, the organism is intersex. Okay, meta males has the ratio of 0.33. Meta females has the ratio of 1.55, and this meta females are weak and sterile. And there is a switch gene, that is A sex has a switch gene. which directs the female development. The sex lethal gene is located on the X chromosome. It has two states of activity, that is, it has two types of activities. If the other X chromosomes and autosomes are regulated by this sex switch gene. If it wants the femaleness, it will on. If it is off, that is SLX gene is on a particular or that is autosomal chromosome, it becomes female. If it is off, it becomes male. And the Y chromosome in Drosophila is only for the male fertility. And the X chromosome was discovered by Henking in 1891 and Y chromosome was discovered by Stevens in 1902. So different doses of X chromosomes and autosomal sets and their effect on sex chromosome in Drosophila. Here the phenotype is given and the number of chromosomes, number of autosomes and the sex ratio is given. So the first one is you will see it from this tabular column. You can get any 1 mark question. So please go through this tabular column and you can get a clear idea using the sex ratio, that is the sex index, the number of X chromosome by number of autosomes. Here they had given the number of X chromosomes and autosomes. You divide and the answer is also there, but you can work out this and see, and you will get an idea about the different doses of X chromosomes and autosomal sets and how their sex, the determination is done in the Drosophila. Gynandromorphs. So gynandromorphs means their body, in an individual, the body expresses the male characters, some parts, and some parts expresses the female characters. This is a Drosophila. This part, that is this portion resembles the female part, that is the female body, and this portion is the male body. In a single individual, the male and female parts are expressing, then that is known as gynandromorphs. Dosage compensation, Barr body. So in 1949, Barr and Bertram first observed a condensed body in the nerve cells of female cat, but it is absent in the male part, that is male cat. So the condensed body is called as chromatin, and later it is named as Barr body. So what means that is Barr and Bertram has discovered the female cat that is seen in the nerve cells, a special type of body which is condensed, but it is absent in the male. So the condensed body is called chromatin, but later it was named by them as Barr body. This is the dosage compensation. As we all know, the sex chromosomes for female 2X and male 1X and 1Y. How they are compensating? Because the female has 2 copies of X-linked genes, that is, but the male have only 1X. So is there any problem in gene compensation? Maybe. So if how they are creating the equal amount of X chromosome, whether the X chromosome is, that is only one chromosome in both male and female is active, another one is inactive. So the dosage compensation, the female has 2X and male have only 1X, and there is only 2 things. The double the amount of transcription takes place in males, that is 2X genes can be double transcription takes place, or in females one of the X chromosome can be inactive. By this only the dosage compensation is fixed. So, and another person, Mary Lyon, the bare bodies are represented and as inactive chromosome, an inactive chromosome which in female becomes tightly condensed, that is coiled into heterochromatin. A condensed and visible form of chromatin is seen, that is the Lyon's hypothesis. So the bare bodies are inactive chromosomes, they are tightly condensed in the female which is called as heterochromatin. This is the Lyon's hypothesis. They are visible also. So the number of bare bodies in a cell was 1 less than number of X chromosomes. So the two in a cell there are 2 X chromosomes. One becomes the bare body and another one is the X chromosome. So like that also it can be have. The XO female doesn't have bare body because there is only one X. So by this they are compensating the dosage level between the male and the female sex. So the number of bare bodies follows, that is follows n minus 1, that is the rule, the number of total number of X chromosomes present. So for example, if there is 2 X chromosome means 2 minus 1, one chromosome is active and one is the bare body. So like that they are calculating, and this is the Lyon's hypothesis. Haplodiploidy in honeybees. In Hymenoptera insects such as honeybees, ants and wasps, the haplodiploid mechanism of sex determination is common. That is, in this, the sex of offspring is determined by number of sets of chromosomes that receives. So the honeybee lays so many eggs. So only the fertilized egg will become the queen and the worker. Unfertilized egg becomes the drones. But it means the male has off number of chromosomes. So they are haploid because they are formed from the unfertilized eggs. Okay. So unfertilized means only one gamete will be there. So it is haploid in number. But the females have double the number, they are diploid. So hence the name came it as Haplodiploidy. The male is haploid and the female is diploid. The combination haplodiploid sex determination, the name comes. And in this diagram you can see the diploid, that is the direct diploid female becomes the queen, and other haploid egg, that is haploid eggs which are fertile, becomes the woman, that is becomes the worker bee, and the haploid, the direct haploid fertilized unfertilized, that is direct haploid unfertilized egg becomes direct haploid egg becomes the haploid egg becomes the drones. That is here from this diagram you can see the diploid queen becomes the queen. It is after fertilized also it becomes the queen, and the haploid eggs which combines with the haploid sperms and they becomes the worker, and they are fertilized one, they are hatched from the fertilized eggs. But the haploid eggs doesn't have any contact with the sperms, they becomes the worker, that is they becomes the drones, that means male. So this mode of sex determination facilitates the evolution of the sociality. How they are socially working. The honeybee is the best example for social being, that is sociality. So only one diploid female becomes the queen and lays the eggs of the colony. So only the queen bee is going to lay the eggs, and other worker bee is also female, no. So they also they contribute their reproduction to reproduce the eggs of the queen bee and to nourish the eggs and to nurse the bees. That is known as kin selection. The queen constructs their social environment by releasing the factor called, that is by releasing the hormone. So the while laying the eggs itself, the queen releases a hormone which suppresses the worker bee to for not to fertilize. So that is because of this, the queen only lays the eggs and produce only one queen is there, and the worker bee is nourishing the eggs and the bees and the drones are doing for the fertilization, because drones is haploid, but queen, that is the queen bee has diploid. So through this it is an haplodiploid sex determination is seen in the some of the insects like ants, wasps and honeybees. Thank you for watching the video. If you like the video, like, share and subscribe for Science Easy Tech channel.